From Microbe TV, this is a special immune booster recorded during the Society for Leukocyte Biology conference at Michigan State University in October 2024, starring Cindy Leifer and Brianne Barker, and featuring Amy Hise from Case Western Reserve School of Medicine.
Today we're joined by Amy Hise.
Hello.
From Case Western Reserve University, and we're really glad to have you here. I was particularly interested in talking to you because you work on some yeast, fungi,
Yep.
and how the immune system recognizes that. And that's not something we normally talk about. But before we talk about your research, can you tell us a little bit about how you got interested in science?
Well, I think even as a kid, I was always interested in science. Like I was always the one who wanted to do the experiments, wanted to build a solar oven with my dad, wanted to watch like medical shows or science shows. So I think that was always there. But I did actually consider for a while going into the arts. And that was my big decision in terms of going to college was, did I want to go more science and engineering or did I want to go into the arts? And I decided that arts could always be in my life, but, like science and engineering, without the training, it's kind of difficult to work in that area. So I ended up, when I went to my undergrad, I ended up majoring in biomedical engineering, where I was one of the few women at that time in my track. So it was really kind of an interesting experience. But I had worked in labs doing research since I was in ninth grade. So I had, I'd gotten through my contacts with my dad. I had gotten summer positions working in research. And in college, I worked down in labs at the medical school. So when I graduated, I knew that's what I wanted to do. I wanted to work in a lab. And initially I worked in industry. And then after I went back to school, then I went to medical school, and then the rest is sort of history. I've been, doing research ever since.
So it sounds like your father played a huge role in your development in science.
Absolutely.
Was he also in science?
So my dad was a mechanical engineer.
Okay.
And he worked in the aerospace industry. So when I was growing up, of course, I'm the eldest of four of us, and he was always my hero and my role model. And it wasn't actually until I was an adult that I've learned some of the absolutely awesome stuff that he did when he was, working as a younger man. When we were very little, he was working at Martin Marietta, and he actually has a couple of devices that went on the Apollo space missions. He designed a device, I'm not sure I'll remember all the details, but had to do with fluid mechanics and how the fuel could stay where it needed to go or could flow where it needed to go. And without that technology, they would never have gotten off the ground. So it's pretty cool to hear his stories about that time.
That sounds really inspirational.
Yeah, really, really cool.
Definitely.
So you mentioned going to medical school and sort of then going on after that. So what type of research did you sort of start your career with as a med student or immediately after med school?
Well, it was interesting because before I went to medical school, I worked for a company that made blood-derived therapeutics. So I was always interested in sort of the translational aspect in human health. I got a master's in public health. And then in medical school, I wanted to do clinical research because I had only done bench research up to that point. So I paired up with an endocrinologist who was looking at thyroid function in cardiac patients. And then when I came to do my residency, which I did in Cleveland, in internal medicine, the chair of the Department of Medicine had heard about my research interests. And at the end of my intern year, called me in his office. I was so sleep deprived. I was half thinking he was going to tell me that there was some issue and I was fired or whatever, because you're so tired. I had no idea what was going on, and he seemed very serious. But what he was inviting me into the office was to talk about an opportunity to study HIV and tuberculosis in a patient cohort in East Africa. So with some trepidation, because I had never traveled internationally like that, but it seems so interesting. So that's what I did. So during my residency, for both my second and third years, I spent time in Uganda. The Case Western has a big TB research unit there. And I was paired with a PI who was studying what happens, with co-infection. And that was my first introduction into sort of how the immune system interacts with two different pathogens or interacts with a pathogen in the context of other microbes being present. And I just found that whole concept so fascinating. So my initial work that I did as a fellow, as a research fellow after my training, was in tuberculosis. And I was looking at macrophages and how they interact with tuberculosis. And I was just starting to get started on that process when a lot of the TB researchers left and went to a different institution, including the PI of the lab that I was in.
Oh.
And I had a little baby, and my parents lived in Cleveland. I was not really in a position to move. So it was really gracious of the, so I was on a training grant, and the director of the training grant gave me time to interview other PIs and other labs and really spend some time looking for a good fit. And in the meantime, I had heard this really fascinating talk by David Underhill. Mm-hmm. It was at the TB 2000 meeting in New York City. I was there. I actually had my baby with me, and my nanny had come with me. And he was talking about toll-like receptors. And in my training, we had learned that the innate immune system was this rudimentary, kind of not important part of the immune system, something that was like an evolutionary remnant.
Yeah.
And so his talk was the first thing that I'd heard about like specificity in terms of how the innate immune system recognizes and is activated by different compounds. So I thought it was the most interesting thing. So I wanted to study innate immunity. So I went back and I, was talking to different PIs in Cleveland. And I found a lab that wasn't studying tuberculosis, but was studying filarial parasites. And a paper had just come out that mentioned toll-like receptor 4 in the context of this endosymbiotic bacteria in the filarial worms. So Jim Kazura had given me the opportunity to, spend some time pursuing this, see if anything could come of it. And I actually ended up getting a KO8 award on that very topic.
And so this is an endosymbiotic bacteria that's recognized by, say, human TLR4 and not the filarial TLR4.
Well, so it's very interesting because as part of that process, so I had to learn how to grow the worms and, learn about the bacteria. It's an obligate endosymbiote with the worms, which is really interesting. There's a whole field of people who study Wolbachia bacteria in the context of other systems, and it's prevalent in insects, as many people may know. Yep. So I went to some meetings on Wolbachia, and I went to some meetings on innate immunity. And at one of those innate immunity meetings, I met Doug Golenbach, who was kind of like, I was a little bit starstruck, but he invited me to come out to his lab. He was really interested in this whole Wolbachia filarial story. And, he had a lot of reagents and a lot of expertise, a lot of great people in his lab. And that was, we were really trying to replicate this paper showing this TLR4 activation. And all I could see was toll-like receptor 2. So it took longer to publish because I just wasn't convinced that, what I was seeing. But it probably was, endotoxin contamination of the original preps that activated toll-like receptor 4. And, with Doug's lab and his expertise and some of the other people in his lab, we were really able to see that it was toll-like receptor 4. Toll-like receptor 2, not toll-like receptor 4. So it was a really cool project. It was really interesting. And I was interested in filarial parasites. I was really interested in taking it to the next step. It was difficult, when you're at that early stage of kind of transitioning from being a trainee to a independent investigator, you're so reliant on the support system around you. And I was having a hard time finding support for clinical trials to really go in the field and test if we target Wolbachia, what happens in patients who are infected with filarial parasites. Other investigators did that. And, some of those trials showed that if you did target the Wolbachia bacteria, you can clear the filarial infection much faster. So I do feel like my own, research moved the field in that direction. I always regretted that I wasn't able to do the field studies, but, I didn't have the experience to do those field studies. And, you need to have a site that's set up and have somebody who's already made those connections and has all the personnel in place on the ground. It's not the same as doing a clinical trial or doing human studies in the States when you're, working in developed, they call it the
developing world.
Developing world. It's not really like, what we call it anymore. But countries that don't have the same infrastructure or have a different, research infrastructure or a different political system, it takes a lot of time to navigate that. So in the meantime, I had gone to a joint lab meeting where it was with an investigator in the dental school who was talking about the microbiome in the mouth. And I had already learned about the microbiome through conferences and colleagues, but I was really fascinated to hear about almost like biowarfare that was going on, at the junction of your immune cells and the, commensal bacteria in your mouth and a pathogen that might be trying to come in. And how, these different organisms utilize the host response to either augment their own growth or suppress a competitor. And I just thought that sounded so cool.
Really cool.
And I naively thought that I could develop a mouse model of oral candidiasis or thrush to be able to look at some of these questions in the context of these innate immune receptors I was studying. It turned out to be much more difficult because mice are not normally colonized by Candida, Candida albicans in particular. But I developed this mouse model, and that was actually the work that my first R01 was based on. And meantime, I had formed a very close collaboration with somebody who had worked with Doug Gullenbach and now was an independent investigator, Kate Fitzgerald, who's a good friend. And Kate was looking at inflammasome activation. So my R01 was both the innate immune receptors and then also the inflammasomes in the context of mucosal candidiasis. So that's sort of how I got into that area. And then more recently, with the emergence of this drug-resistant, multi-drug-resistant pathogen, Candida auris, it seemed natural to kind of look in that direction. And so it's been difficult to develop mouse models for Candida auris. Most of the models involve IV injection of the Candida auris in a very lethal model, and I'm trying to look at mucosal responses. So it's been difficult, but we have a model, and we're seeing some really interesting things come out of that. And one aspect of that I'm really interested in is if we look at the interaction of the microbiome and the commensal bacteria with Candida auris. One of the things that we know from human infections is that people don't come into the hospital for the most part with Candida auris infections. It develops after they've already been admitted to the hospital. And so it's really, I think the average length of time from clinical studies that people are hospitalized before the Candida auris infection is detected is about 9 days. So, people's immune system is suppressed, or they've had surgery, or they've had powerful antibiotics that alter the microbiome that allow the Candida auris to overgrow. Some people are carriers of Candida auris. That's increasingly being seen. And then it's also nosocomially spread from people who are carriers or from patient to patient in a hospital setting or in a nursing home, that type of environment. So for most, most people who are walking around healthy, Candida auris is not a health threat. But when your immune system is altered or suppressed through, medications or through illness, then you're more susceptible. And we're just starting to tease apart, how it's different than other species of Candida and how the immune system is activated, and what are those factors that really make people susceptible.
Are there particular members of the microbiome that seem to be associated with susceptibility?
There are potentially, though we haven't published that work yet, and this is pretty early. I'm working with a collaborator. I work at the Cleveland VA Medical Center, and that's actually my research right now is supported through the VA system. So the Veterans Health Administration grants, you really want to focus on health issues that affect veterans. And so this is definitely one. We have, veterans who are in situations where they're going to be susceptible. And my colleague studies antimicrobial resistance and infection control in hospital systems. So it's really interesting to have that kind of collaboration where you come at the same topic from a different viewpoint, right? Because infection control is really important, not just for Candida auris, but for many other pathogens, and especially some of the drug-resistant pathogens that we are really hoping that we are not going to see increasing in prevalence, or that we're not going to get colonized with in the healthcare setting. So, it does appear that there are certain bacteria that might be a marker for susceptibility, or it might actually be that there's an interaction between the microbes. We're not quite sure yet.
Cool.
Yeah.
I was interested, you mentioned a couple of times your child and taking him to the meeting and things like that. Can you talk about what have been challenges or what advice you have for young women trainees who are going into science?
I think it's really challenging, both in medicine and in science, to navigate parenthood, and not just for women, I think for men to a certain degree as well. I mean, my situation, I'm a single mom. I've always been a single parent. And, that's part of the reason I stayed in Cleveland, because my parents were there and I had family support. I think, there's no right time to have children. I went to medical school with people who had kids. I went to medical school with people who already had kids and people who got pregnant and had kids during that part, and also residency training, which can be, very long hours and, it can be a very difficult period to get through. So, they managed, they navigated. I think, if you wait until all of your ducks are in a row and your career is set and you're financially set and, you've got all the support you need, you're going to be like 50 before you are, in that state. So you can't wait. You have to just have kids when it's right for you. And, do the best you can. It's really important to build a community of support, whether that's family members, your own immediate family, or, your chosen family, pairing up with other parents so that you can share childcare responsibilities. Just having that network in place is really important. Also, I think it's really important to realize that your career is not linear. It's not like you are starting and you're taking these regular steps, climbing up through your career, right? I mean, there are some people that probably have a career that look like that. Most of us don't. And so you'll have different chapters in your career, and there's different chapters where you're going to be focused on different things. Early in my career, I was fortunate to have a career development award from NIH, and that mechanism, that funding mechanism, allows you to have devoted time to your research. In fact, it requires that you have a certain minimum % effort focused on your research. And as an early stage faculty or clinician, that's really important, because otherwise you're going to be pulled into teaching responsibilities or clinical work. So having that focus was really helpful for me. And my days did not always look like an 8 to 5 day or an 8 to 6 day or an 8 to 7 day, right? There would be days where, I was in early, I was working in the lab, I would leave early and take my kid to the pediatrician or, something like that. And then after he went to bed, I would be working for 2 or 3 hours in the evening. I often had a very productive writing period after he went to bed at night. So, I had to make sacrifices in my life to have that kind of focus, but it did allow me to have time with my son when he was really little. Then when he was older, I was traveling a lot more for meetings. There were some meetings that I took him to. Having my parents close by, he had a bedroom there. He was comfortable there. I could just give them the dates and he was taken care of while I was traveling, which was a blessing. I also took my parents and my son with me on trips. So, there was a toll meeting in Ireland, and it was my parents, my dad still talks about that meeting because we tagged a family vacation on the end of it. So there's times that I've traveled with them. There's times that I left him at home. And I think it's also hard as a parent, especially a parent of young children. You feel like your presence with them is so important, and you don't want to miss any of these developmental milestones. I mean, my son is 25 now, and looking back on it, I mean, I kind of think he needed me more when he was like a teenager or preteen. When he was a little kid, anybody can take care of him. They can give him, feed him and read him books and, play with him and put him to bed. His needs were met. But as an adolescent and a young adult, he needs my input as his mother. And we have a very close relationship now. I don't feel like the time away from him that there was any, cost to that. If anything, he benefited from a closer relationship with his grandparents, and I benefited from being able to pursue my career and network and just have that break from parenthood. And even at 25, he calls me all the time. He discusses, problems that come up in his life. I see him all the time. So I feel like, I did the best I could at times. Sometimes I felt like I was treading water. Other times it felt, like everything was going really well. And now looking back on it, I don't think I would change a thing.
That's great. The big question is, did you take him to the library to look at to look at books and stuff?
Oh, absolutely. Actually, he also, when I was doing research, sometimes, when I was hands-on in the lab, I would have to do weekend work or evening work. And so he was very familiar with my offices. He had books and crayons and toys to play with. When I had to go feed the filarial worms and change their media every day, he was tagging along with me. He's engineering minded. He's very scientifically minded. And, I think that exposure to science was really important. I think it's also really important to expose your kids to working parents and the challenges that they face.
Yeah.
So what advice do you have for trainees?
In terms of
a successful science career?
I mean, I think the most important thing for anyone at any stage in their career is to know who you are and know what your values are, your professional values, your scientific values, and your personal values. Like, know who you are, know what you're interested in. I mean, it's not going to do you any good to pursue an academic career if you're more interested in, something like this, science, communication, or media. Likewise, there's industrial careers are really interesting. There's a lot of resources there. It can be a very different way of doing science. So there's so many different career options. I think, really look broadly to see what's going to fit best with who you are and what you want to do. The other advice I would give is to really stay focused. It's super easy to get distracted by all the cool science that's out there. And maybe, you're working with a mentor or a PI that, wants you to kind of pursue something else in this direction, or this might be interesting. You go over here. I mean, it's really interesting it's really important in the beginning to stay focused on, a narrow topic. You can pick a field and then pick what's going to be your niche in that field, because you want to build your network of collaborators. You need to build your reputation. You need to publish in that area and, start to get funding. And all of that takes time and focus. Maybe after you've had some success in that, you can have a side project that kind of looks at something, different. But it's easy to get distracted. And I think if you get distracted too many times, you're kind of starting and stopping over again, and it's very inefficient. I mean, that being said, if you really like that, sort of broad focus, there are areas of science that you can do that, bioinformatics or, some of the newer techniques that we have where you're really looking at something very broad. So I think that's really important. And I think getting advice from people who have gone through the path is important. For me, that was really critical. And some of my mentors and some of my advisors were not in my institution or in my department. They were people that I met at national meetings or international meetings. And that can actually be helpful to get the opinion and the advice of somebody who's not in your current system, can see things from the outside and give you good advice. You're going to run into roadblocks. You're going to have stumbles. You're going to have setbacks. Resiliency is really important. And I think it's easier to be resilient when you know what your focus is and you know what your values are and you know what you want. Because then, when something happens, you are able to make decisions to still get where you want to go in following a different path. It's easy to get discouraged when you hit those roadblocks or when things don't go well, things don't work out the way you think they're going to. But, you should know and recognize that there's more than one way to get from here to there. And so you might have only focused on this one path, but there's going to be multiple ways to get to where you want to go.
That's great.
That is great advice.
Well, I think that we can wrap that up here.
Great, thank you.
Thank you so much for coming on.
It was a pleasure to talk to you.
It was really great to talk to you.
Yeah, thank you so much.
Thank you for doing this.
That's a special episode of Immune and Immune Booster. Questions and comments, please send them to immune@microbe.tv. If you enjoy our work, please support the science shows of Microbe TV at microbe.tv/contribute. Microbe TV is a 501c3, so your donations are U.S. federal tax deductible. Our guest today from Case Western Reserve School of Medicine, Amy Hise. Cindy Leifer is at Cornell University. Cindy Leifer on Blue Sky. Brianne Barker is at Drew University. BioProf Barker on Blue Sky. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious.